A very blissful morning to the assembly of respected honorable dignitaries, respected faculty members, resource persons, and our beloved students. Welcome again to the second day of our 4th annual national conference on food informatics, opportunities and challenges. Today, we will have 3 technical sessions. In technical session 1, we have the opportunity of broadening our horizon of understanding through the lectures of Dr. J. Srinivasa Rao, Deputy Director and Scientist E, National Institute of Nutrition, Hyderabad; Dr. Debobroto Bera, Associate Professor, Department of Food Technology.
Good morning, one and all. Please be silent, please, because as yesterday in my keynote,
Please keep quiet.
please take your seat, Noma.
I request all participants to kindly keep silent.
Yeah, good morning, one and all again, Director, and after the who are sitting, including faculty and the invited experts, my dear students. First of all, I would like to heartfully thanks to Professor who introduced me such a beautiful way. Again, I shall try to approach you and try to reach you by the way how the dietary diversity in Indian context is useful and in what sort of the things as a students, that means in my keynote address yesterday itself, I discussed many issues regarding the shortfalls in India in terms of the dietary diversity and the malnutrition issues yet to address, especially from the student community, the way which you are pursuing nutrition dietetics as well as the food technology relevant courses which is very much need to give awareness at community level. In this concern, all these things actually yesterday we discussed. There is no need to discuss much about, and actually the chairperson already gave me caution that I supposed to take only 35 to 40 minutes. No. So in these regards, I shall try my best to reach you. I think till yesterday, we discussed a few things, how the India is facing malnutrition issues. Among that, the overnutrition and obesity is also one of the major problems nowadays, but especially in the non-fragile countries, including India. So, coming to the dietary diversity, in what way the dietary diversity that we need to maintain at community level, and what would be the benefit, what are the resources, and what are the applications the students supposed to come up to in these dietary diversity complexes in Indian context. If you could consider the dietary diversity application, it is a qualitative measures of food consumption that reflects household access to a variety of foods and is also a proxy for nutrient adequacy of the diet of individuals. I hope that you can understand, because dietary diversity is nothing but of the, when we are taking the food substances, whatever the essential foods are there which they are entering into the body, they should have to scattered and enrich to the individual body complications, body applications and the metabolic activities. Here, if you could consider the classification of foods which are very much relevant to the nutrition, so based on functions of these foods, again here these can be considered into three categories. One is energy yielding foods, as we know that, so the first category which we end on in this energy yielding food is carbohydrates, which is immediate energy yielding food into the body, then fats. Among that, in my yesterday's slide, one of the slides I mentioned, India is one of the countries now we are coming up with the latest data on trans fatty acid levels. Earlier we don't have the data on the trans fatty acids. Now the NIN, ICMR NIN, we have been associating with the CDC USA and we are generating the data on trans fatty acids next to the USA, because we have already data, especially in foods, only on MUFA, that means monounsaturated fatty acids and the polyunsaturated fatty acids, but the trans fatty acids data is yet to publish from ICMR NIN. And coming to the other food, bodybuilding foods, as we know, the macromolecules are considering under this category, that is proteins, and the protective foods, again vitamins and minerals, based on the nutrients which are supporting molecules to the biological system, are again the macro and the micronutrients. Here the again vitamins and the proteins. So what are the strategies and how would these strategies will help to maintain the balanced diet at the community number? Here when we are talking about the food strategies, you ask them to sit, no? Okay, right. Actually, when we are talking about the strategies in terms of the biodiversity, the two things that would like to discuss purely depends on the availability and accessibility of the individual essential molecules after intake. So here the proper strategies are, majority of them are preventive and cost effective, sustainable and income generating, because the strategies when government is coming up with policies which needs to implement at community level, those would be relevant to the cost effective, sustainable, how the strategies are sustained as well as the income generating. Like many strategies are there, I think there is no need to discuss much. So in addition of these strategies, I would like to give you few examples actually in what way they are coming up and how they are giving the priority, as I mentioned in my previous slides also. Now, in Indian context or the global level, many new food groups are coming. Many new food groups are coming. Among that, the microgreens are one of the food groups. In addition of that, the organic foods and the GM foods like genetically modified foods. So in what way these kind of the differential foods are playing an important role to maintain the food habits and the nutritional aspects as per the RDA when compared to the normal foods which are already available. These are the kind of new food groups which slowly the Indian population has been adopting. These are actually, the microgreens are a tiny particles which we have only 2 to 3 leaves, and the harvesting time is also very less. It's more almost from 8 to 12 or 14 days only, these microgreens are. And coming to the nutritional aspects, these microgreens are playing an important role when compared to the conventional foods available in the society. Because as we analyzed these microgreens, one of my students has done her PhD on these microgreens, and we published a couple of papers on these. These are the usefulness of microgreens when compared to the conventional foods available in the society. If you could attend these microgreens in your daily life twice per week, actually whatever the non-communicable diseases and the malnutritional effects which we discussed earlier in the previous day, most of the things can be able to eradicate while attending these microgreens our daily life. She is my student actually. She worked it on these microgreens and she published a couple of papers. She prepared actually using the microgreens many recipes. And we analyzed these microgreens. The next one is fortification strategies, because nowadays the policies has been developed from the government of India. These fortification strategies also can be used to increase the content of certain trace elements, especially because the Indian population, when compared to the developed nation, the Indian population are severely suffering with especially trace element disorders, trace element malnutritional disorders. So this is one of the approaches which will, if we will increase the fortification activities at in the foods what we are attending in our daily life, there are chances to address the malnutrition activities at community level. Of course, many issues are already being under the progress. And what are the benefits of these diet diversification activities? The first one is preventing diseases. Once if you will come up with the fortification foods, a diverse human gut microbiome is normally a good sign and well correlated with an individual's overall health. Whatever the diversified foods are there, these kind of the foods are maintain balanced activities in the human biological cycle to control the malnutrition issues by diversifying the foods. Actually, in today's world, because of many losses in diet diversity, and actually the diets which are fed by the essential nutrients are also playing an important role in terms of the gut microbial activities, especially the human studies. And the second one is, the second benefit by the diet diversification is essential nutrient intake. Essential nutrient intake, that is also when compared to the recommended dietary allowances in the food. So if you would have to enlarge the quantity of essential nutrients through the diversification activities, we may have a chance to reduce these essential malnutritional activities at a community level. Of course, healthy weight is also one of the parts that I don't want to talk much about these things. And the NIN, ICMR NIN has come up with, recently we came with a balanced healthy diet plate. If you could see here, this is the balanced healthy diet plate, because like the developed countries of US and UK, the other developed countries, just 2 decades back, they came up with a strategic plate, that means meal plate, which consisting of more than 50% of the diet is occupying with green leafy vegetables and the essential nutrients which are not much harmful to the body, like based on those recommendations, that means the WHO and UNICEF FAO guidelines, the NIN, ICMR NIN, we also came the recent my plate concept, and we are encroaching to the society, which is very much essential to come up with such kind of the foods. This plate is consisting of more than 50% of your daily plate. What the NIN ICMR recommendation is telling is, it must fill with green leafy vegetables, and the some kind of the fruits, and the rest of the 25% is cereals, and rest of the 25% is again some fat as well as the oil and such kind of the things. If we will have to attain, this is not much difficult also to attain. Then only it is possible to maintain the good health in terms. This is the urban India actually my plate concept, and this is the rural India my plate concept. Yes, we know that the rural India people are attaining more than the recommended dietary allowances of only cereals. It is per day more than 400 grams of the cereal has been taken by the rural India population, as we know, because the policy is given by the government of India. So what are the interventions at community level to address these malnutrition issues? Since from 1900 onwards in India, we came up with many actually intervention programs to address the malnutrition issues what yesterday we discussed, like the iodine deficiency as well as the anemia Mukta Bharat, and in addition of that, the vitamin, number of vitamin A deficiencies, everything has been already initiated. But to implement at community level, still we are facing many issues to implement at community level, because lack of awareness about the fortification programs, as well as to maintain healthy activities and healthy individual activities at the community level. If you could see here, the first initiation problem, universal salt iodization for iodine deficiency, has been initiated in 1962 in India. Actually, the Indian government has came up with this policy in 1962. But the program has been strictly implemented from 1983 onwards. 1983 onwards. Of course, still there are a lot of shortfalls. Even the government, the policies, though we developed many activities to eradicate this iodine deficiency at community level, but still to implement these policies, when the government is changing, they have been coming up with a lot of changes. Of course, this problem is also there still in India, even after 75 years of the independence country. And the second one, as I mentioned, the recent NFHS 5 data, yesterday already I mentioned, saying that anemia is one of the important issue at community level, whose the people are severely suffering. Still, even the recent NFHS 5 data, it is mentioning that more than 57% of the adolescent girls, as well as the reproductive age women, in addition of the children community, are severely suffering at the community level. So to address this problem, actually, iron and folic acid supplementation for anemia has been initiated long back in 1970s. But still, even after 70 years again, we have more than 50% of the 57% of the anemia is still in the society, that the people are severely suffering. Of course, this program was initiated in 1970, but particularly the initiation and to address these activities has been initiated from 1991 onwards. So the impact of the government of India is taking many issues to address this problem, while attending through the meal process as well as the fortification activities, but still the problem is as such at community level. Then the other problem, as I mentioned, our country is standing at rank number 2 in terms of production of all green leafy vegetables and fruits, but still the vitamin A deficiency is one of the major issue at community level. Because as per WHO recommendations, if the problem is 0.5%, it is a public health concern problem. But still in many of the states in Union India, we have already more than 0.7%, like Madhya Pradesh and Kerala, some other Jharkhand. These states are still severely suffering with this vitamin A deficiency. The other important issue which I would like to mention here is, as the vitamin A cannot directly intake through the diet. The only problem is it is available from the secondary metabolic products like the carotenoids. So many studies in terms of the carotenoids are telling is carotenoids is the oxidizing compound again, carotenoids. Even at 25 degrees itself, the all carotenoids can be oxidized, and we cannot expect as usual the quantity of carotenoids here. And many studies, even myself and one of the scientists from CFTRI, Dr. Vimala group, we have done many studies on this carotenoid bioconversion. At global level, the studies are projecting that 4 molecule of carotenoid is equivalent to 1 molecule of vitamin A, 8 molecules of carotenoid into 1 molecule of vitamin A, like 25, 15, 21, 28, even recent paper published from US, actually they mentioned that 31 molecules of carotenoid is equivalent to 1 molecule of vitamin A after conversion takes place into the efficacy activities in metabolic activities. But our studies, what we found was, the other problem in Indian context is, we are the people giving much priority to take the tasty food rather than the nutrient retention. So we are adapting towards the tasty food only rather than the nutrient retention. And our study was also actually mentioned that while cooking, using different cooking practices at Indian context, the losses of these oxidation molecules ranges from 5 to 72%. 5 to 72% of the vitamin C and these oxidative molecules like carotenoids and other essential molecules are going to be losing while preparing the food substances with our culinary practices. That is one of the major problem at Indian context, which we are still in the problem of this vitamin A deficiency, which even the government is also trying to address this problem while through the supplementation activities. But at community level, again, there are a lot of shortfalls. The other important issue here is food fortification policies. Of course, there are many issues. Yeah. So food fortification policies, of course, in India, the food fortification policies has been started in 1950s, but it has been implementing from 2000 onwards. We may expect the fortification foods to become, and the Prime Minister is also, the present Prime Minister is also very much giving priority on these food fortification policies. So these are the policies, and coming to the strategies how to implement for the dietary diversification. When we are talking about the strategies, simple things if we will adapt at community level or in our home level, at kitchen gardens or wherever it is possible, we can able to address these malnutrition issues. Home and school nutrition gardens, that is not much difficult. Because now if you will go to the rural schools or the rural villages, we have a lot of spaces available. But the only thing is again awareness. So ultimately, whether we have to implement the biofortification foods or organic foods, whatever the kind of foods, the only thing is community mobilization is the first thing that we need to take initiative. Unless we have to take initiative to mobilize the community, it is very much difficult to address even these malnutrition problems. Though the government is coming with many policies like fortification, genetically modified foods, and the policies which they are implementing like iodine policies or anemia policies, many policies which have been coming up, but even then to enrich to the public and mobilization is one of the important challenges in India actually. And actually the other important thing is just the agriculture field is very negligible field in India. Even including the educated people, we are also not giving much priority to this agriculture discipline. Nutrition sensitive agriculture activities we must have to initiate, and we actually at community level we have to change their behavior activities. We have to change their behavioral activities, then only these kind of the things will be applicable. Farming system for nutrition study, because we are not giving much priority to the nutrition. That is also one of the important aspects we need to address this problem. And need for multidisciplinary center for food activities. Multidisciplinary centers, rather than what we are doing it at present. If we come up to multidisciplinary centers in terms of the preparation of food grains, that also one of the approaches which we need to address these policies. Actually, so as far as the expert concern, the overall India needs to promote holistic food-based approaches around this double iodide salt as well as the behavioral change communication to not just address population-based malnutrition disorders, but also promote healthier lives as long-term and sustainable strategies, which is very much required. And in addition of these strategies as well as the things which we need to address at community level, what are the other concerns that Indian population needs to concern. So the major things which are reflecting to the agriculture field is increasing levels of carbon dioxide. That means this is relevant to the again environmental fact, one of the environmental factor, because day to day the pollution activities at community level has been increasing. That is the reason which is playing an important role in the society. Because of these activities, the temperature has been rising. It is affecting on the yield as well as the nutrient content which we are coming up with, the even new organic foods also at community level. As well as the other important issue is, so year to year, the UNO is actually giving caution to the countries like India, the groundwater level has been decreasing. This is also one of the major challenges actually in Indian context. And many other causes are there to address these things. Actually, what's supposed to do? As a student community, what we're supposed to do? Actually, the challenge is to consider our strategy very carefully. It must include a quantitative evaluation of the problem. Nutrition gaps must be calculated, but we do not have correct intake data so far. Though we need to calculate the nutritional activities after intake of the meal, but in India, like the other Asian countries, so far even we don't have a particular data to calculate the intake amount of the energy. Because I'll give one example here. Recently in 1917 again, the ICMR NIN, we came up with IFCT book, as most of you know about the IFCT, Indian Food Composition Tables. In that food composition tables, actually we covered more than 200 foods which are being consumed by the Indian population. The nutrition exploration, everything has been incorporated in that book. And now, when we are considering the population-based surveys or studies, the energy calculation is being based on the raw food intake only. But this is not the method. We have to come up with the data on the cooked foods also, as well as the after cooking, the yielded food data also need to be incorporated when we are calculating the energy levels. Then only what exactly the energy gap and the nutritional issues to be needs to address at community level, we will come to know. Of course, if you could see the other developed countries like fragile countries, they have a data. Even a portion of recipe data also they have been calculated and giving directions to the public to sensitize and to give awareness about their healthy concept. So this kind of still the gap is there in Indian context. We need to come up to address these gaps. So again, what the RDI, recommended dietary intake, is telling. This is again the NIN-ICMR published data in 2020. We came up after 10 years. The recommended dietary allowances, if you could consider an adult man which he is the aging of 60 years. So as far as the study is concerned, he is supposed to attain 2,700 kilocalories per day to maintain the healthy life. 2,700 kilocalories per day. But instead of that, he is meeting only 2,000 kilocalories per day. Just 2,000 kilocalories per day. If you could see here, the gap is almost 700 kilocalories he is supposed to meet per day. As well, the adult woman, if she would have 55 kgs, she is supposed to attain 2,200 kilocalories, but she is attaining only 1,700 kilocalories, which is close to 500 kilocalories energy gap is there. Like if you could consider all the age group, like pregnant women, lactating mothers, and the different age group from 1 to 17 years, rather than the demand which they are supposed to meet in terms of the kilocalories, they are not properly meeting the actual intake due to the many regions, and this is the energy gap which we need to address. This is the data published, and of course this is computed energy. So in this context, for student purpose, what are the areas they're supposed to come up with the research? You see here the micronutrient research, especially the students who are pursuing the food science technology as well as the nutrition and the dietetics courses. The micronutrient research, especially among the micronutrient research again, the main shortfalls, especially in Indian context, so far the studies are mentioning is zinc deficiency is one of the prevalent, as well as iron, as I mentioned, the anemia problem, vitamin A, folic acid, vitamin B12. Actually, the government is coming with many policies to address these issues in terms of the fortification, even though there are a lot of shortfalls are there to enrich into the community level. So then the new areas of research can be considered into basic, clinical, program research and policy research. I don't want to take much time here. Actually, in the basic research, again I mentioned that biomarkers, micronutrient biomarker studies also need to come up, especially the students if they could think in terms of this micronutrient biomarkers research. Actually, that is the good area which can easily able to give you the platform, and also whatever the objective which you are coming up, even the many government agencies are also ready to accept your objectives if it is relevant to the community-based approaches. Then government like ICMR, many government agencies are also ready to give the fund to address these issues. So currently the available biomarkers represent steps too far along the disease process, because whatever these biomarkers studies has been done in Indian context long back, so, and the present situation depends on the upcoming new food groups like genetically modified foods, again whatever I call the microgreens and the other organic foods. The studies earlier done was so far to address these kind of the disease burden at present in the society. And the proteomics and nutritional genomics, actually your college is already working it on that. And the other areas which very much required is updating for required establishing accurate methods. Though we have such a vast population and a large country, but so far the research methods which we established at community level is still 10 years behind than the developed methods what they are using it for the research data generation. This is also one of the new approach that we need to come up. And the other important area which the students need to sensitize is bioavailability and the bioaccessibility of the micromolecules. Because our daily intake, the food substances, though it is the cereal kind of food or the millets kind of food, whatever the food, we are attending the quantity more than what the body is required. But many studies are telling the bioavailability and bioaccessibility of the essential molecules after digestion takes place are not properly reaching to the targeted organs. Again, that is also one of the encroaching activity in Indian population we need to think seriously. And the RDI, as I mentioned again, the clinical research, the bioavailability trials, actually efficacy trials, and the important activities in terms of the microbial studies are also very much rich in Indian population. And the program research, the other important issue which I would like to mention is translational research on food fortification. Because still majority of the students are looking towards either the molecular biology studies and the genetical studies, some kind of the advanced studies that they are looking at. But this is one of the important area which the students really needs to think, because the translational research in terms of the fortified foods, how they are giving priority to the community level is also one of the approaches. And large scale epidemiological studies, we don't have actually data on, though only few organizations are working on this epidemiological study data at community level across the India, but still there are a lot of gaps are there that we need to address while doing the epidemiological studies and generating the data at country levels. And so to enhance these kind of the programs, we need to have public-private partnerships also. Because even recently, like the Institute, City College, and even the other organizations, they are taking the initiatives to interact with the existing organizations at national level. And even not only the City College, I have been visited many the private colleges and private universities. They are also ready to interact with the established organizations in terms of the student exchanging and faculty exchanging programs, which such kind of the approaches definitely will give a positive opinion to enrich and to address these malnutrition gaps at community level. And the last but not least, the other important area is policy research. How the strategies needs to develop at community level to maintain the multiple encroaches to reach while addressing these malnutrition activities. So while discussing about the policy researches, economic analysis is also one of the important part. So we have to do the balance kind of the approaches to address these policy researches. Of course, what are the projections that India is looking after to come into the society? Like the other developing nations at global, India is also one of the countries which recently from 2 decades onwards, just from 2 decades onwards, we are taking a much priority and giving more interest to generate the data on the nutritional aspects, and giving priority as well to do research studies, especially on underutilized foods. Underutilized foods, that these are called as, again, of course they may be comes under the genetic foods, or some indigenous foods also coming under this underutilized foods. The country has also utilized the evolving knowledge and invested in evidence-based intervention programs, especially to improve food and nutrition security for the citizens of the country, as well as ensure that the ongoing food supplementation programs, as I mentioned already, Penny, only few I mentioned, as well as nationalize and improve ongoing nutrition interventions aimed at prevention, early detection, and effective management of under and overnutrition problems. Of course, right now, the overnutrition is one of the burden to the country when compared to the undernutrition. Of course, the undernutrition country population is facing problems since 75 or more than 100 years onwards. But the overnutrition population, day to day, month to month, the data has been increasing like anything. So despite, these are the challenges actually, despite the positive trend, India remains burdened with an actual unfurnished agenda of undernutrition and communicable diseases, as well as the non-communicable diseases also it is there, especially yesterday we discussed about the diabetes and the other issues. Addressing this dual burden of over and undernutrition is critical to achieving, unless there is a cooperation and there is a coordination among the all organizations, it is very much difficult to address all these problems. But coming to the research prospects, still we need in-depth research. In this area actually, still we need in-depth research. We need to work it out and generate a data on markers which we need to come in with a promising data, especially on markers which are playing an important role in terms to eradicate these malnutrition issues. So in this context actually, large-scale trials on different doses of micronutrients again, because whenever or whatever the issues which we are talking about, this undernutrition and overnutrition, micronutrients are playing an important role, as we know. But the changes actually recently, an increasing interest in assessing the bioaccessibility and bioavailability. As I mentioned yesterday also, we have already sufficient quantity of iron as well as the other components every day we are attaining through the meal. But to reach after digestion, whatever the essential molecules which we are thinking, they are not properly reaching to the gut biomarkers, gut biodata, as well as the targeted organs which needs to address these malnutrition issues. That is one of the problems. And the other issues here are the microgreens as well as the green leafy vegetables and the fruits. When compared to the other foods, if you could consider, the bioavailability and bioaccessibility is more when compared to the other food groups here. And what are the fields, especially in food science and technology, in terms of the research? These are the actual more 4 or 5 fields that the students need to address. Food microbiology is one of the upcoming area. Definitely it will help us to address whatever the pathogenic studies still the Indian population is suffering, and safety aspects also to be addressed by this food microbiology area. Food engineering and processing also, because that's what we need to have interact from each department to the other department, then only it is possible to come up with the sort of promising data. Food chemistry, this is playing an important role in terms of the data generation, which is very much essential, and need to generate the data on actually the cooked foods also, nutrition. These are interrelated subjects, nutrition as well as the food chemistry, as well as the food biochemistry, and the nutrition is also one of the promising areas that we need to address. And the sensory analysis studies are also important, because majority of the universities or majority of the colleges, they are coming up with sort of recipe preparations, and which the recipes are prepared by them really are having good approaches to meet to the community demand. But the only thing is, they don't know actually how to evaluate through the sensory evaluation studies. So to address these problems again, we need to come up with a sort of training programs among with the organizations. That means the developed organizations as well as the developing organizations. Then only the student community as well as the young faculty will get it then little bit approaches how to address these problems. And the other core research areas are the global food security is one of the problems. Of course, this is again pertaining to India also. Microbiome as well as the microflora studies, which are very much essential at Indian context. Gene expression studies, which now the gene expression studies are also seriously thinking towards the nutritional content and the energy metabolism studies as well. The inflammation studies, especially in women community, women population, the inflammation studies also likely need to come up, and the aging, because day to day in India also, as I mentioned yesterday, of course India is no young country, because more than 40 crore of the people are below 35 years of the age. But the aging is also one of the problems. Geriatric population percentage in India also has been increasing year to year, that we need to address what are their problems and their issues in terms of the nutrition is comparatively the younger generation, their issues in terms of the nutrition is different. We need to give the solutions for them also. Then the biomedical engineering. Biomedical engineering studies also one of the other important promising area that we need to come up. And nutrition education is more important rather than all these things. Unless we have to educate the people at community level to address whatever the so far the activities we discussed, it is very difficult unless we have to educate the people sorry, at community level. And interdisciplinary and sorry, cross-disciplinary collaboration at global level as well as the country level is also helping to emphasize and to generate the data at community level. So last but not least, if you could see this slide, the street foods as well as the junk foods at community level. So here the street foods and junk foods, especially in India, has been occupied by two sectors. Two sectors. One is organized sector and the second one is unorganized sector. So according to this Red Sea Consulting Agency report in 2020, India is one of the major countries which the street foods and junk foods are being consumed by the population. And you see, this is the per capita income which we are spending to attend these street foods and junk foods. This is one of the promising area when compared to the other areas right now which we need to address. Because these street foods and junk foods long back has been developed by the developed nations. Now they are maintaining a good healthy practices. And simply they thrown out these kind of the street foods and junk foods on to the developing countries as well as the underdeveloped countries. So we are exclusively every day, every 1 out of 3 we are attending either the Golgappa or something is there, no, what is that? Pav bhajis and the pizzas, burgers, whatever it may be. But they're not hygienic in terms of the nutritional composition is also not good, and in terms of the nutrition composition, the heavy metal composition is more. This is my actually recently I proposed this project to the health ministry and it got approved actually. Hopefully in couple of years I may come up with the promising data on these street foods. These are the selected street foods in my study which we are going to be addressing the issues. And coming to my department activities, as you know, so we have the world-class facility in NIN. So out of this, we are attending the proximate principles which consisting of 9 parameters here. And this is the book which I mentioned already, Indian Food Composition Tables. We came up with 2017. And so when we are considering this book for nutrition analysis, the entire India has been divided into 6 regions, and the food has been, the same kind of the food has been collected from each region, and we transported to the NIN, and after compilation of the food, we analyzed. So the other important issues which I would like to mention here is, so assessment of energy and nutrient intake. This is one of the major objective of food chemistry department, what we are doing it. Non-nutritive food components also we are attending to generate the data, and we are providing the training programs, especially in the areas of analytical areas of standardized sophisticated analytical instruments like UPLC, as you know already yesterday you mentioned that. In addition of that, LC, MSMS, ICP, MSMS, GC, MSMS, of course protein analyzers, many instruments are there which are helping to generate the data on this. And the first time in India we are generating the trans fatty acid data in biological samples.
soon in 1 year we may come up the entire data which we need to publish next to USA. But here the data is still, when we give, so far the generated data is, when we compared with USA data, the trans fatty acid levels again in the Indian population are much higher than any other countries, including US. The trans fatty, that's what the CHD and CVD cases are also more in India than the USA, even the other developed countries. That is also one of the programs. And the other important area which we are working is particularly vulnerable tribal groups foods. That means indigenous foods. We are working it on that. And the non-communicable diseases, of course. In addition to these non-communicable diseases, the chronic kidney disease is also one of the prevalence in tribal population, which we are working it on this area. Still, even though we are coming up with the promising data on different kind of the foods, still we have many gaps in the food substances available in the country. So we are planning to have generate the data on these food graphs, and these are the instruments which we have: ICP-MS, as I mentioned, and HPLC, GC-MSMS, which we are using it for the trans fatty acid as well as the MUFA generation, LC-MSMS, protein analyzer, of course, biochemical analyzer, all these things. And this is the study, as I mentioned. This is pertaining to only Telangana state, where my study I am working. This is the data from Arogyasri report in 2020. You see the CKD cases in different districts of Telangana. These are the CKD cases. Then once if we consider at a national level, we cannot imagine what could be the percentage of people are severely suffering with the CKD. And as I mentioned yesterday, this is the mahua, indigenous flower, being collected and consumed by the tribal people all over India. And we extrapolated the nutritional composition. We found that the macromolecules of protein and the dietary fiber values are very much favoring when compared to the conventional foods which we are using in our daily life, as well as the iron and calcium levels are also very much high. So after sensitizing the nutritional composition, actually we prepared a recipe in terms of the laddu. This is the mahua laddu, and we have done the efficacy toxicity studies on this mahua. Then we started one intervention study. We have conducted one intervention study on 3,000 tribal pregnant women, and we received the promising results in terms of the improving of hemoglobin levels in the 3,000 tribal pregnant women who are attending in this study in intervention progress. And of course, we are closely associating with the Andhra Pradesh, Telangana, as well as the national governments to promote these kind of the activities. This mahua, now the Telangana government is coming with this mahua as the nutrition kit to distribute to the pregnant women soon. Actually, recently they released a GO on this mahua recipe also. Yeah, thank you, Varandal, for your patience. Yeah, it's okay, sir, no problem.
Thank you very much, Dr. Rao, for your highly illuminating and fascinating lecture. Dr. Rao, in his lecture, highlighted the way malnutrition and micronutrient deficiencies are emerging as serious health problems all over the country. He also highlighted how to combat these problems. He talked about promotion of microgreen culture, fortification-based intervention programs, and he also suggested what an ideal plate for the day should be. And then he elaborately discussed how to combat micronutrient deficiencies, especially iron deficiency, folate deficiency, iodine deficiency, and vitamin A deficiency. Then he talked about food fortification strategies in India. He talked about the strategies, current status, and also the challenges being faced by such fortification programs. And then he talked about the new horizons of research that should be carried out to combat the problems of not only malnutrition, but different micronutrient deficiencies. And at the fag end of his lecture, he showed how his laboratory contributed to, to devise an innovative food from indigenous resource, and that was Mohua Laddu. I wish he could bring some pieces for us also.
Definitely I will send you, sir.
He forgot. Anyway, now the session is open for discussion for a brief period. And I shall be happy if students and researchers ask some questions and interact with sir directly. Any of the students, please. Yes, please. Please stand up, tell your name, and ask your question very neatly.
Sir, my name is Sumon Maiti. First of all, sir, thank you for your amazing lecture. Sir, as we know, India still have a huge number of people belonging to BPL category, and India have a high number of homeless people. They are living in footpath, busway, and railway station. These people are a major part of anemia and other food-related problem. So, there are any activity that ICMR are taking to treat them?
Yeah, good. Actually, what his question is, majority of the people in India are living below poverty line, and those people are particularly reflecting towards these malnutrition activities. Of course, the government of India is coming up to address all these issues, not particular community. Even the state to state, the programs are different, depends on the ruling parties. Actually, we should not supposed to talk about the political issues here. But when the policy has been designed by the central government, majority of the states in India are not being applicable, applying those kind of the policies to address these kind of the issues, because again the political crisis. But hopefully, rather than that, as a student community, my message from here is, if we will give awareness in terms how to address all these problems at community level, that is the best solution rather than the policies which even the government is coming year to year.
Thank you.
Yeah.
Good morning, sir. I am Sneha from Midnapore Autonomous College.
Yeah, ma, please.
Sir, in one slide, you mentioned that one of your students work on GM food product.
Not GM food, microgreens.
Yeah. So my question about that portion, sir, do you really support that modified, microbial food product? Because in India, I have studied that there was a controversy. Many of scientists said that it is causes harm in future because it process is modification from microorganism. So those genes by food intake our body. So in future it may harm or it may causes cancer or tumor. So what is your opinion about this?
Yeah, fine, good question. But here I think you need to sensitize basically here two things. Microgreens are actually natural ingredients, first of all. If you have to grow, you can grow even here also, no problem. So there is no harm. We are not using it any kind of the pesticides or the other things for their growth. So they are natural ingredients. After harvesting, after 8 to 10 days, just simply we are using it for and we are mixing it with any. We can able to mix with any kind of the foods which you want to prepare. And the other end, what you said is right. Some genetically modified foods. There is a controversy at national level. Still, the big debate is already it is running on the genetically modified food, because majority of the genetically modified foods so far, what they are shooting towards the inducing cancer activities in the community. Of course, the government is supposed to take initiatives whether they can come up with these genetically modified foods or not.
Sir, those microgreen fruit, what is the benefit or why should we choose microgreen food?
Okay, good. So microgreens means that's what the community is severely suffering with the essential malnutrition problems like anemia, if you could consider iron deficiency, iodine deficiency, thyroidism. Like many other issues are there. When compared to the intake of conventional foods available in the market, if we can able to grow microgreens in and around our home area or kitchen garden, wherever it may be. So these foods are addressing while giving the availability of iron, calcium as well as protein and dietary fiber is the supporting activity. That's what they are benefit in our study which we mentioned already. I published already a couple of papers also on these microgreens. Thank you.
Sir.
Good morning, sir.
Yeah, ma'am.
My name is Aika and come from Midnapore College Autonomous. Sir, my question is the plant MOA is only present in Telangana?
No, it's not like that. Yeah. No. Here also it is available, huh? Sir, here also it is available? Okay. Wherever, but
Okay,
it is a seasonal flower. Seasonal flower. Available from February to April, May only. Yes, scarcity also. But the problem is this flower being collected by the tribe people, they are using it for the alcohol preparation after fermentation. 48 days fermentation, 48 hours fermentation. That is the problem.
Thank you, sir.
Sir, as usually, okay, Masit. If we will intake that alcohol even after 48 hours fermentation, as such in the purified form alcohol, if you will intake also, that is good for health. But these tribal people, what they are doing, to get the kick, they are adding it ammonia as well as the urea during this 48 hours. That's what majority of the kidney cases actually we found in Andhra Pradesh and Telangana. Of course, this may be in the entire India also, I hope.
Thank you, sir.
Yeah.
Any other person? Is there anyone else?
Yeah. Yeah, indigenous foods are many, but so far we don't have proper data. Actually, which indigenous foods and which kind of the component is working against the cancer in human gut relation. We don't have proper data. I think the studies are now coming even. Your colleges also, I think you are doing it towards the direction I studies. Yeah. Okay. Yeah, Kaushik.
Sir, I have no question, but I have request to you and NIN and ICMR. We just see the present bioavailability of the any type of nutrient. Suppose the iron recommendation for us is a various age level. We know, and iron present in the food.
We know. Yeah.
We know, but how much iron by particular food in our body bioavailable, it is not unclear. This, if you or your NIN something do for the particular food, how many iron present that, how many intake and bioavailable and in our blood entry, that is very important. Then we choose particular food, particular amount for the diet.
Okay, very good suggestion, Dr. Kaushik. Here the thing is, though many studies at global level, okay, why we should talk about the global level? In Indian context, many studies has come up to address and to quantify the exact quantity of iron is available in terms to the cells. But the major prevention here is, as you know, the phytic acid is the major preventive agent. The studies, even one of the department in our biofortification division, the biofortification division group is working it on that, how to prevent, how to reduce this non-nutrient, especially the phytic acid, and they are trying to calculate what exactly the quantity of available iron after digestion takes place. Most probably, I think in 2-3 years, we may get it, the solution for this, to calculate the available iron level after digestion. And the one more thing I would like to, with the chairperson's permission, one more thing I would like to tell you here, the institute is open for any kind of short-term projects. Since from last year, after pandemic, so we opened for the short-term project students, especially the post-graduation as well as the PhD students. You please try to utilize. In addition of that, we have already 2 post-graduation students at master's level, MSc Nutrition and Dietetics, and the one more is MSc in Sports Nutrition. If any students are interested, you please contact your faculty members, and the PhD students, you better to visit once NIN or CFTRI under the student exchange program, then you will get more knowledge. Then where rightly you are working, not only here, any other college students or any other organization students, better to utilize these kind of the opportunities. Thank you very much. Thank you, sir.
Thank you, sir, very much. And our director and TIC, sir, they are seriously thinking over your invitation, and we hope that we'll be sending some project workers in short time. Thank you very much. Now I would like to invite our second speaker, Professor Keshav Chandra Mondal. Before Professor Mondal starts talking, I would like to briefly introduce him to the audience. Professor Keshav Chandra Mondal is the head of the Department of Microbiology in Vidyasagar University. He is an eminent scientist who works on microbial enzymes and their practical applications. Production of fermented probiotic foods, ecology of gastrointestinal microbes, and development of antimicrobial substances from herbal sources. His research output is prolific. He has already utilized grants, 12 research grants received from UGC, DRDO, CSIR, and DST of Government of India. His research output also includes as many as 12 PhD, and it is indeed a pleasure to inform you that our TIC sir, Dr. Kuntal Ghosh, pursued his PhD studies under direct supervision of Professor K.C. Mondal. Professor Mondal was elected a Fellow of Physiological Society of India and also a Fellow of Biotechnological Research Society of India. He is a member of Indo-Hungarian Educational Exchange Program, and he is also the recipient of Ramendra Sundar Sinha Memorial Award. I invite Professor Mondal on behalf of the entire hall to start his deliberations and enrich us.
Good morning, everybody. Particularly, I offer my gratitude to the chairperson of this session, the director of this college, my distinguished scientist, my dear student. So what Professor Srinivasan sir mentioned, I will start from that point. And what many issues raised, that we consume many things, but that not bioaccessible and not utilize our body. Why? I will highlight that portion particularly. Very interesting new topic of research. And this part of the research mainly focused after 2006, when human genome project completed. Human genome project initiated from 1993 and terminated 2003. Initially, it was the idea that there are present more than 1 lakh of genes, but finally when people assess, they find only 25,000 genes. Next question arises, where the remaining 75,000 genes? And at that time, they give him this gene as the junk gene. Later on, human microbiome project initiate. That is the forgotten organ of our body. I will discuss some point on this related to this. And from 2006 to 2016, after 2016, many things has highlighted. Human genome only contains 25,000 genes, but the so-called microbes which remain in our body, that having more than 1 lakh genes. Okay, what I stated that human is more prokaryotes than eukaryotes. Whatever supplementation happened, I will discuss some points. So this is the usual concept of the healthy diet. You have the idea that healthy diet mostly composed of adequate quantity of macro, micronutrient and energy total calorie. There is also many definition from the WHO that the diet must protect malnutrition and simultaneously it should protect non-communicable diseases. Non-communicable diseases, what mentioned earlier that regarding cancer, heart disease, obesity, these are belongs to non-communicable disease. And according to British Nutrition Foundation, a healthy diet should provide the right amount of energy from food, drink to maintain energy balance. These are some definition. And next come the micronutrient deficiency. You have idea that we also know that micronutrient, mostly the vitamins and mineral which needed very little quantity, and deficiency leads to many health consequences. Many disease, and mostly 2 points very important. Productivity loss. Direct productivity loss, that means lifespan reduced, therefore productivity loss. And second is the indirect productivity loss that leads to poor mental development, schooling, increased cost of healthcare. So you have idea that government spent a lot of money for the healthcare system. So it is a burden for the government. And government initiated many programs for the fortification. We have the idea, iodine fortification. We all mostly use the iodized, double iodized. But what is its fate? I will give some results. Till now, this excess iodine leads to many consequences. That portion remain in the shade. People only added different micronutrient, but excess consumption, what will be its fate? That is the interesting point of discussion. Government initiated many program, and for particularly integrated child development scheme, government spent around more than 20,000 crore. Similarly, for maternity benefit, Pradhan Mantri Matru Bandhana Yojana, Janani Suraksha Yojana, government spent around 10,000 crore. This is huge. But till, what is the condition, present status? Government initiated many programs that mostly reach into the grassroots. This is the condition. Till now, iodine deficiency 70%, folic acid deficiency 37%, most interestingly iron deficiency 54%, similarly vitamin B12, vitamin A, vitamin D 61%. Next question arises, why? If we consume regularly iodized salt, okay, so why we are till now deficient of 17%? So why micronutrient supplement program is not so effective? There are many causes. Major cause is the lack of knowledge or research on the behavior of the body system. For example, same medicine not exhibit similar effect in the 2%. Similarly, this fortification system is not so effective all community, all people. So why? So people mostly not giving emphasis what I stated earlier from the 2006 to 2016, our gut flora, gut microbes, this is called forgotten organ. Forgotten organ. Amra bhulei gechi, this is a particular organ. And its activity is more than liver. We have the idea what is the function of liver. It is the metabolic total machine. Their collective activity. So it's 9-meter length containing trillions of microbes, but they are very teeny. Their quantity not more than 1 to 2 kilogram, and 95% are the bacteria. What I stated earlier, the genome composition. Here only 23,000 genome in the human, whereas gut microbiome more than 10 lakh genes. And the total surface area of the gut, more than 2 tennis courts. And if you roll these microbes one by one, gut bacteria one by one, it will cross the earth more than 2.5 times. Individual, whatever bacteria in our gut is remain, if we make a row, that will cross the globe more than 2.5 times. These are so metabolically active, and many these are the classification on the basis of their phyla. There are major 5 group of bacteria: firmicutes, bacterioids, actinobacteria, proteobacteria, and vericomicrobia. And so these are the major phyla, and their distribution depends upon many factors. Their distribution depends upon the many factors: geographical location, food habit, many factors affected the composition of the gut flora. This is one table that highlight the adult of USA containing around 59% firmicutes, whereas in case of Indian that is 62%, remaining are the difference. So they have many functions like nutrient competition. Earlier, just one question arises: if the food containing that amount of iron, is this bioavailable? That means our body will get that quantity of iron. I will give answer that no. That completely depends upon your gut flora, gut microbes, their biological activity. And they produce different vitamins, and very interesting, 10 to 30% of they can extract energy. For example, if we needed 2400 kilocalorie per day, 30% means 800 calorie extracted by the gut flora. Remaining 1600 calorie extracted by our digestive enzyme. Is it clear? So imbalance of this group of microbes leads to many consequences I will discuss. They have many other functions. They produce lot of enzyme. For example, simply, we only secreted, our body system synthesized only alpha amylase, okay, for digestion of the starch. But a food containing different types of sugar, it may contain resistant sugar, beta sugar, many types of sugar. Suppose we consume more than 2 kilo of food, but our stool weight not more than 50 gram, 500 gram. Next question arises, how the mixture is so-called decomposed, digested. That is due to, one of the portion is due to microbial activity. We have the simple experience. For example, if we suffered by one acidity, one problem one day, acidity means that is the microbial reaction. Our bodies secreted acid and gas produced by the microbes, not our body. So, when acidity happened, pH altered, what thing happened? Our digestive enzymes somehow their activity reduced. But the microbial bloom happened. And that leads to changes of acidity, gas, etc., etc. And now perhaps you have idea that 3 short chain fatty acid is the major communicator in between gut lumen and human body. What I stated, that gut microbes are the beneficial, but how they exert the beneficial effect to the host. Until day, whatever research completed that highlighted that 3 short chain fatty acid, one acetic acid, acetate, butyrate and propionate, these are actually synthesized by the microbes, and these short chain fatty acid then diffuse into the body, and our all organ from brain to every organ containing receptor for this short chain fatty acid, and they modulated from our emotion, behavior to everything. And if we have so-called balanced microbes in our body, if we have balanced composition of microbes in our body, what thing happen? We have no need to consume any external vitamin, as because most of the vitamins more than RDA synthesized by the gut flora, including biotin, cobalamin, folate, niacin, pantothenate, pyridoxine, riboflavin, thiamine. Gut flora synthesized adequate quantity of vitamin. We have to not needed from the external source, if we maintain our gut microbial ecology completely. If the ecological balance disturbed, ecology means what I stated earlier. Perhaps you've seen that Indian having the Pacific group of microbes in different proportion, American having the different group of microbes in different proportion. If the balance somehow change, altered, then leads to many diseases. This disease can be broadly divided. This condition is called dysbiosis. And if you have a good balance, that condition is called eubiosis. So dysbiosis means alteration of the group. Some group become bloom, some group decrease. For example, obesity, what stated earlier, that is due to alteration of gut microbial composition. You see that gut microbial ecology changes leads to many intestinal disease like inflammatory bowel disease, IBS, celiac disease, CRC, CDI, like intestinal disease. But apart from the intestinal disease, these are the extra intestinal disease like obesity, type 2 diabetes, metabolic syndrome, lupus erythematosus, allergy, gut-brain axis disorder. These diseases are due to alteration of the gut flora. What Sir Srivas had mentioned earlier, that junk food, what is its impact, major impact? Junk food is called energy dense, nutrient poor food. Energy dense, nutrient poor food that actually altered gut microbial composition. So there are many causes of the there are many factors are related to the alteration of the gut microflora that initiate the so-called colonization of the gut microflora. Colonization of the gut microflora. Initially we have the belief that our so-called when baby form develop, the so-called our fetus is free from microbes, sterile. But the concept has changed from 2017, 18. Many research publications arise that colonization of the gut flora begins before birth. And that is the very interesting thing that we'll discuss later. But thing is that there is simple evidence. Obese mother and generally their baby is the obese. Lean mother, their baby is lean. So-called alteration of the metabolic pattern, fingerprint, start before birth. So next comes into the iron deficiency. I will give some idea about our research. Iron deficiency, this is huge. Sir mentioned our RDA level and many approach till now developed, many approach. What sir stated, fortification food, dietary pattern change. As a microbiologist, I can say that they may help up to certain limit. But major important point is that whether if you have adequate good microbes in our body that can assimilate that iron, I will give some idea. So whenever iron supplementation is generally leads to many common symptoms like constipation, gastric irritation. In case of infant, that is also related decreased growth, impaired cognitive development, and increased diarrhea. That thing happens. That is commonly whenever any individual consume the iron tablet. That is a common phenomenon. And if this so-called supplementation program is, I mentioned earlier that so-called fortification, this is the 4 decade that started 4 decade, but just you see the data, 2005 and 6, 2015 and 16, what change happened? What change happened? Till anemia prevalent, this is the condition, So if the fortified food or by means of any way are so applicable, so beneficial, so healthy, I will give some idea that these are the states, mostly central and eastern states are where the anemia is more prevalent. Next come into the physiological aspect. We consume iron by means of 3 products. One is the inorganic, heme and ferritin. This is the endocyte, that means around 15% iron absorbed in the duodenum and more than 27% iron absorbed by our colon, that is the microbes dense zone, and in the middle other percent. So this is the process. Initially iron Fe3 reduced to Fe2, that Fe2 enter and again our system convert to Fe3, and in the middle this iron captured by the one protein transferrin and that delivered into the target organ. This is the process. So major problem happen, so-called microbial interference, that is this upper portion, this is the reduction. Initially reduction followed by oxidation. Reduction facilitated by the microbes. Microbial surface protein actually reduced. Therefore they control the somehow reduction process. So there are some point very important. We observe that most of the micronutrient deficiency remain in the rural area, not in the urban area. Mostly micronutrient deficiency prevalent in the rural area. Next, this is my opinion. Question is why? As because we depends upon some plant food. Plant containing many, what Srinivas sir maintain, one phytate. But apart from the phytate, this polyphenol, tannin, calcium, they actually trap this micronutrient, trap. Therefore, these are not bioavailable. Similarly, another is the oxalic acid. Egg contain one protein called phosphatin that inhibit the iron absorption more than 30%. On the other hand, these group of food, particularly one question arises, why developed country not suffered by this sort of deficiency? The answer is simple. They mostly depend their food culture based on the meat and fish. That is the major source of bioavailable iron. They adequately consume, but our rural India, so-called economic condition, there is some barrier. Vitamin C support, there are other some of acid, they also support. So this group, from these to these are supportive, these two up top, these are somehow inhibitory. Inhibitory means if we consume plant-based material regularly, then they can prevent all sort of ionic imbalance. They can trap all sort of ion. Just you see our civilization, early civilization. Most of the people, our civilization is more than 6,000 years. So earlier people, they prefer to consume flesh. That means that is the major source of all sort of micronutrient. Next, come into the gut flora in the aspect of the iron metabolism. That gut microbes regulate overall balance of the micronutrient. What I stated earlier, that if we maintain the so-called balanced gut flora, that is very helpful, that can synthesize all sort of vitamin. But apart from this gut flora synthesized, what mentioned one point is that a group of enzymes that hydrolyze so-called phenol, tannin, etc., phytic acid, then ion become bioavailable. Apart from this short chain fatty acid, this is the target of research. Those are working in this field, this is the target of research, how we can improve the quantity of short chain fatty acid. As because, in the next slide I will discuss, they have the potent effect, not the brain or other tissue. They can extend the surface area of the endocyte, therefore facilitate movement of the ion. Second thing is that they actually they create some sort of membrane potential change that leads to rapid diffusion of the ions. Okay, am I clear? So short chain fatty acid, acetate, butyrate, propionate, these are the main target molecule. They communicate all sort of ionic movement. And third is that siderophore-like material, siderophore. This is the iron trapping compound that's synthesized by the bacteria. So this siderophore, they can trap different ion, including divalent ion, zinc, magnesium, they can trap. And they can easily enter into the endocytes, therefore no need to other way. Okay, so two important things, one is the short chain fatty acid, second is the siderophore like compound. Question happened, Suppose there is iron, 100 number of iron. This is the micronutrient. So microbes also needed this micronutrient. When they get this micronutrient, they will be multiplied. Okay. When they multiply, their free iron content is very less. But some group of bacteria, they can trap, initially bind with this iron or zinc or magnesium. Therefore, they protect iron from they protect iron from the diffusion into the other group of bacteria. And this trapped iron then diffuse into the cell. So, this is the role of the short chain fatty acid in the different layer. I am not, somehow we have to speed up. So, when the iron balance, what I mentioned that if we consume iron, suppose supplemented, what thing happens? Some portion will be assimilated. But if your body system not so-called supportive, what thing will happen? This iron will support growth of pathogenic organism. What sort of symptom appear in the so-called child or pregnant woman? That due to alteration of the gut flora, child diarrhea, that is due to multiplication of a group of organism. So group of organism can be multiplied due to iron supplementation, and finally what thing happen? Reactive oxygen species, that means oxidative damage happen in the intestine, and our immune system also affected. Next, microbiome based therapeutic. This is the new concept. Just you see some group of probiotic organism or fermented food, these can protect the gut flora. They can prevent the dysbiosis. So if you consume regularly probiotics, the concept is now changing. That local probiotic is now the new concept. Local source probiotic, as because what group of probiotic developed by the Japan, that organism not so effective. When that come via the Amul or Yakult into reach to the Midnapore, that is somehow so-called effectiveless. Therefore, the concept is the local. Local means if you consume Medinipur doi, Medinipur dokan theke je kono doi khao, that is more healthier than the Amul doi. The thing is that microbes make simple adaptation. Previously they adapted in our body. Therefore, they can comfortably multiply within our body. So, group of microbes, you have the idea, the definition of the probiotic. These are useful to prevent the anemia. These are useful to prevent the anemia. As because how they can prevent the dysbiosis. Therefore, they are supportive for the gut flora. So these are the alternative what I stated earlier also. So we have to rethink personalized medicine, personalized therapy. This is the new concept. এক অস্ত্র সকলের কাছে কাজ করবে না. New concept is the personalized medicine. That is happening. আমার জন্য which is beneficial for me. So this is some, I want to show some sort of research very fast. We have isolated 2 good probiotic organisms after rapid screening, and thereafter we applied these group of microbes for the support of the anemia. We made some induced anemia by treatment of the benzene. This is the standard method. And after that, we make different group and added our probiotics. We tried in the animal model, which will be most supportive. So before that, we screen whether the siderophore, what I mentioned that they can able to produce. This is the level of siderophore that can be synthesized during course of the growth, and this siderophore can be synthesized in presence of or little availability of the iron. Little availability of the iron. That as because each organism having some sort of behavior. So we have first tested and thereafter we applied for 8 weeks. We observed that many parameters, blood morphological analysis, we have measured different parameter, hemoglobin concentration, MCV, MCH, different parameter. We found that our organism somehow effective. Thereafter, we have studied the iron content in the serum by means of by studying different parameter. That is also somehow supportive. Next, iron level regulatory protein. There are some local hormone. They are control iron absorption. We have studied and they are somehow beneficial. And we studied the transport system, studied its transport system, whether these are express or not. This is somehow beneficial. And we studied by the qPCR level, that means quantitative PCR, the intestinal microflora. We have studied that whenever iron, suppose control, this just you see that, this is the quantity of lactobacillus, this is the control. Whenever we added supplemented iron, their quantity decreased. Okay, this is the curve of the anemic condition. But whenever we supplemented our probiotic, their quantity increased. So how they increase? We mentioned that they maintain a favorable condition. They secreted siderophore. Therefore, they can trap the iron and that assimilate into the body. And finally, concluding remark. So malnutrition, just you see the picture, particularly I draw this. So whenever malnutrition happens, then increase the quantity of opportunistic pathogen. When again supplemented, then increase the quantity of true pathogen. But this is the normal or our healthy condition. So both condition, deficient condition or so-called dose, overdose concentration, both are detrimental for the gut microbes. Both are detrimental for the host. So what is the solution? Next portion is this is the solution. Personalized microbiome based intervention strategy. Personalized, that means initially it should be examined what quantity microbes are there. Considering this, likewise whenever we visited any doctor, they first analyze TC, DC, ESR like that. After that, they started the medicine. Similarly, this is the true good time to so-called, if we try to control this sort of deficient state, we must have to focus our forgotten organ. Its function is more than liver. So personalized microbiome based intervention strategy. So how it could be possible? Through our local food, local traditional food. This is the major idea. We have to again return to our traditional system. Yoga, personalized, this sort of again come in. Ayush system again come back. So likewise we have to make this sort of intervention. So lastly, there are in fact two things, science and opinion. The former based knowledge, the latter ignorance. So my dear student, the literature, particularly UG, PG student, I have an earnest request. So whatever written in your book, that is good. What stated earlier that fortification that is beneficial, true beneficial. So answer is, there is a question mark. So we have to rethink rethink. And lastly, one point I want to mention why I am shouting for the traditional condition. One island, that is the Okinawa Island in the Japan. জাপানে একটা দ্বীপ আছে ওকিনাওয়া দ্বীপ, সেখানে মানুষদের অ্যাভারেজ এজ মোর দ্যান 100. How they maintain? They depends their own culture, depends their traditional food culture, activity. Therefore, their lifespan increase. So, what stated earlier that one business, another health effect, junk food, fortified food. As per my opinion, there are many resources very close to you, nature created. We have to rethink about the resource. Okay, thank you very much.
Thank you, sir, for your illuminating lecture. Professor Mandal initially highlighted what a healthy diet means and the impacts of micronutrient deficiencies in diet. Then he highlighted the direct and indirect losses in productivity due to malnutrition and micronutrient deficiencies. Then he highlighted the tremendous physiological importance of gut microflora and the consequences of disturbance of gut microflora. Then he elaborately discussed the role of gut microbes in the process of iron absorption and making the absorbed iron bioavailable. Then he talked about his laboratory's contribution in isolation of 2 bacterial strains that significantly can modify iron metabolism in animal body. Finally, he talked about personalized microbiome-based intervention strategies. Thank you, sir. Now the topic is open for discussion. I would like to invite our students or students from other institutes to interact with sir directly. Yes, please. Do you like to ask some question?
Good afternoon, sir. I am Keadas, comes from Vidyasagar.
Louder, please. Slightly louder.
Vidyasagar Institute of Health. What substances are present in probiotics that are helps in micronutrient absorption?
I mentioned earlier that microbes is liberated one different type of product, but one of the important product is the siderophore. Okay, this is the polyacronated substance. That can trap iron, and then the total complex come to the enterocyte via endocytosis. This is another route apart from the ferrous-ferric state. Okay.
Thank you, sir.
Major thing is that the condition,
Anyone else? Please tell your name first.
Hello sir, my name is Kanika Patra. I am from Raja Narendra Lakhal Women's College, Autonomous. Sir, my first question is that we have microbes in our body that secrete biotin or other vitamins, but sometimes we take supplement from outside to maintain hair and skin problem. Could their long-term consumption have any adverse effect in our natural microbial system?
That is the feedback inhibition. That is the common thing I mentioned in case of iron, in case of iodine. So hypothyroidism, hypothyroidism, their quantity increase, frequency increase due to continuous consumption of iodized salt. Like vitamin E, if you continually consume, that is some sort of, that is called hypervitaminosis. that is some negative feedback effect.
Or sir, my second question is that how to
Louder,
maintain.
please. Ekটু জোরে।
Okay, sir. Sir, my second question is that how to maintain gut ecology or gut flora or gut microbial balance so that we don't have to take biotin or other vitamins from the outside sources.
I mentioned earlier that if you maintain your gut flora, there is no need to consume any vitamin.
Sir, how to?
You have to maintain. So you have to depend your own food, own culture, own system. Adaptation. Suppose for example, we are staying here. Here environment is quite somehow different. Some day very hot, cold like that. So if you consume regular your local food, local processed food, that is very supportive for your gut flora. That is simple.
Okay, sir. Thank you.
Anyone else?
Hello sir, I am Ruksha.
Yes, please.
So I have a question that why we taking medicines from outside, means personalized medicine, say that.
Not, this is not initiated personalized medicine. This is a common system. But this is the final we have to move. Personalized medicine, personalized therapy. This is a whole.
Means I want to say that Indian people are not. Basically, we are not follow the doctor's prescriptions. We have other
That is-
medication, right?
That is true, but our so-called total medicine, medical science is not so developed. For example, simple. We have no such Indian data of hemoglobin. We have to depend upon the foreign country. Similarly, our blood pressure. Now people saying that blood sugar of the Indian people should be plus minus 50. That means our PP value or fasting value should be plus minus 50. These are new concept is coming as because we depends upon the carbohydrate based food. But the data are mostly from European. Their food habit is quite different. So this sort of data should be generated for the sake of the Indian people. We have no such own data. Own data how it will develop extensive survey. Then come into the mean value. Like that. Sorry.
Please hand over the microphone. Is there any database or the availability in the world
regarding the combined of the microbes in even also the micronutrient combined the commercial compounds nowadays? Is there any report or the others? So far my knowledge, according to the US government and the European government, they developed so many types of the nutraceutical compounds and booming up the pharmaceutical company also. But in the bacteria, in particularly the after beginning of the 2000 or last decade of the 19, we had seen that the probiotics supplementations as a pharmaceutical pills, all the others are almost very much administered along with the any types of the tablets or the pills or the capsule like that. But is there any information regarding the combines of the probiotics and the micronutrients?
That should be. But one thing is that gut microbiome project worldwide started from 2006 to 2016, but Indian gut microbiome project just initiated from the 2020, thereafter COVID period, and we have to wait for another 10 to 15 years. So what is the condition of the Indian people?
We can use the probiotic as our medicine?
That is a good question. We are using, for example, if we depend upon our traditional food culture, there is no need to supplement from outside. For example, panta bhat that contain huge quantity of health beneficial bacteria, which scientifically termed as the probiotic. Okay. Likewise, for example, one point mentioned earlier that the tribal people, tribal culture, okay, and there the traditional food culture is very nice. From the northeast, I also did some work that they regularly consuming fermented food, fermented beverage. That is health supportive, health supportive. If the tribal, what thing happen, I found that culture is somehow they polluted in the sense. For example, we know that Adivasi people prefer to consume on fermented rice, that is the hadia. Okay, but to attract the consumer, what thing happen? Sir, suniye na, very interesting. So what thing happened? Whenever this material selling at the market side, to attract the consumer, they added some sedative medicine to attract the consumer. Therefore, that is the major point, that is the major toxic element behind all sort of problem. But if we visited many extensively surveyed on this pool. There are many tribal groups, they regularly prepared by their own for their consumption. That is very healthier. I did one project and compared that difference between buttermilk, which is available in the market, ghol, and the hadia. Asman Jomin tofat. Bakhor interesting. That is the made up of herbal product, herb, not old ferment. Herb, they use some specific herb for the fermentation.
My name is Sairi Ghosh from Midnapore College Autonomous. My question is, দীর্ঘদিন ধরে অ্যান্টিবায়োটিক খেলে বডি অ্যান্টিবায়োটিক রেজিস্ট্যান্ট হয়ে যায়, তো সেই ক্ষেত্রে কি প্রোবায়োটিক ইউজফুল হবে?
এখন তো ইউজ করাই হচ্ছে, অ্যান্টিবায়োটিকের সাথে প্রোবায়োটিক তো সাইমালটেনিয়াস ড্রাগ। হ্যাঁ, এখন তো অ্যান্টিবায়োটিক ভালো ডাক্তার দিলে আর কি। Our medical system, what I stated earlier, somehow polluted. Okay. তুমি যদি বুড়ো ডাক্তারের কাছে যাও, সে একটা অ্যান্টিবায়োটিক দেবে, তার সাথে একটা প্রোবায়োটিক ট্যাবলেট দিবে। একজন মেরে যাচ্ছে, আরেকটা সে প্রলিফারেট করে যাচ্ছে। ওকে, কারণ এই ইজ হোয়াট স্টেটেড আর্লিয়ার দ্যাট দিস ইজ এ হিউজ বিজনেস। সে অ্যান্টিবায়োটিকটা বিক্রি করবে, আর সো কল্ড অ্যানাদার থিং ইজ দ্যাট উই হ্যাভ নো সাচ ইফেক্টিভ প্রোবায়োটিক। এই যে একটা ড্রাগ বিক্রি হবে, ওকে, সেটা তার এফিকেসি তো টেস্ট করে হয়েছে। আমাদের দেশে রিসার্চ গবেষণার সো কল্ড কোয়ান্টিটি বা পপুলারিটি, আমরা অনেক কষ্ট করে একটা রিসার্চ করলাম, সাপোজ দ্যাট ইজ ইফেক্টিভ, বাট যে রিসার্চ করলো সেই যখন রিসার্চ শেষের পরেই সেই প্রোডাক্টটা বাক্সবন্দি হয়ে কিছুদিন পরে নষ্ট হয়ে যায়। কারণ তার যতই এফিকেসি থাকুক, আমাদের দিস ইজ দ্য প্রবলেম, যে কোম্পানি কিনবে না, যার ফলে মাল্টিপ্লাই হবে না। দেয়ারফর নিডেড ইউ লাইক পিপল, নিউ কনসেপ্ট অ্যারাইজ ইন আওয়ার কান্ট্রি, স্টার্ট আপ প্রজেক্ট, তোমরাই পারো চেঞ্জ করতে। তোমাদের মধ্যে সেই বিবেকানন্দের কথায় প্রত্যেকের মধ্যেই দেবত্ব আছে। আমার অনেক স্টুডেন্টকে আমি দাঁড় করিয়েছি, দে আর গুড এন্টারপ্রেনার, প্রত্যেকদিন ক্লাসে বলে তোমরাই যে প্রবলেম, ছোটখাটো প্রবলেম, কারণ গভর্নমেন্ট এখন প্রচুর সাপোর্ট করছে, তুমি যদি তোমার যে ড্রিম তাকে যাতে রিয়েলিটিতে কনভার্ট করতে পারো, ছোট্ট একটা কিছু প্রোডাকশন ইউনিট। ওকে, কোয়ান্টিটি না স্যার, তার সাথে সাথে ধরুন একটা কোয়ান্টিটি, ইফেক্টিভ কোয়ান্টিটি তো দরকার। আপনি শুধু এক চামচ দই খেয়ে নিলেন, তাতে কি হয়? দেখো আমাদের এই ট্রেডিশনাল ফার্মেন্টেড ফুডের কত তোর মানে হচ্ছে হিস্ট্রি। কোনো পরীক্ষা দেওয়ার আগে বাড়ির লোক কপালের তিলক কেটে দেয়। Why? There is great science, not conventional. আমাদের 6000 ইয়ারের সিভিলাইজেশন যদি কোনো কনভেনশনাল না 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 তা নয় তা নয় তা নয়, that probiotic colonize on your surface, then multiply. Symbolic. জাপানেতে যে হেলথ কেয়ার, জাপানের যে স্কিন কেয়ার সিস্টেম, জাপানের স্কিন জাপানের হেলথ কেয়ার।
Thank you, sir, very much. In fact, we are having more lectures than we calculated earlier. So we shall include another lecture in this pre-lunch session. And the final speaker in this pre-lunch session we shall have another lecture in this pre-lunch session. And the final speaker in the pre-lunch session will be Dr. Devobroto Bera. I welcome Dr. Bera on the stage. At first, Dr. Bera will be felicitated by our students. এই দিকটায় নিয়ে যাও। Dr. Devobroto Bera is an associate professor Dr. Bera is an associate professor in the Department of Food Technology and Biochemical Engineering of Jadavpur University. He had his BTech, MTech and PhD degrees from the same Jadavpur University. His research interests circle around development of food products from marine resources, development of food products from industrial byproducts, enzymology, and ethnic fermented foods. Dr. Bera has utilized research grants from DST Government of West Bengal, DST Government of India, as well as ICMR. He has around 50 publications to his credit in national and international peer-reviewed journals. Today, he will enrich us with his lecture on Bengal cheese, an Indian ethnic food. Welcome, sir.
Chairman and other dignitaries, faculty members, and my students. Topic of today's discussion is Bengal cheese, Indian ethnic food. Basically, cheese, all of we know that now we are habituated with cheese-like food. Suppose 15 or 20 years before, we are not habituated with cheese. But now that is easily available in market and we are habituated for consumption. And one of such cheese is Bengal cheese. So from its name, we know that it is originated from Bengal, that means in Hooghly district. Probably
before 400 years ago, there was one Portuguese colony near Bengal, and Bengal church is also there. And those Bengal Portuguese people, they are habituated with cheese-like product. And at that time, we Indian people or people around this region, we habituated for consumption of whole milk only. But cheese means that is modified chana, basically modified chana, fermented, and different types of modifications are there. So modified chana is cheese. And though we are not habituated with cheese or chana, that time we consumed only whole milk. But Portuguese people, their demand was cheese. So in that time, Portuguese people, they were prepared cheese from horse milk, goat milk, etc., not from cow milk. But their cook, they were prepared this Bengal cheese from cow milk. And so, age of this Bengal cheese is more than 400 years. And this cheese is not popular till now. In 2016, we got this project from DST West Bengal by Bengal cheese. That time, those experts, they asked us that what is the market potential of Bengal cheese. That time we defended that. Now market potential, that is about 20 lakh per year. And then they asked, so 20 lakh per year, and your project proposal is of 18 lakh. So how it is feasible? That time we told that now available Bengal cheese, that is not healthy. At the same time, this is 4 years older cheese. That means ethnic type of cheese. So if we preserve this cheese in healthy way and in packaged condition, in that case, its market value, that is not only national, it is international. There is international market value also. So now there are two, next slide. So it is available in Bengal. Not only Bengal, it is now available in Tarokeshwar also. And there are two types of Bengal cheese. One is non-smoked or normal Bengal cheese, and another type, that is smoked Bengal cheese. Because shelf life of Bengal cheese, that means cheese or chana, that is very less. Suppose maximum 1 week. But if we smoked, in that case, its shelf life is more than 1 month. So there are two types of Bengal cheese. One is normal Bengal cheese and another is smoked Bengal cheese. And with the same line of Professor Mondal, this is a natural product, so it contains different types of microorganisms, both harmful microorganisms, that means pathogenic microorganisms, as well as some beneficial microorganisms are also present in this Bengal cheese. So if we isolate these advantageous or beneficial microorganisms, and if we apply for different food product development or in pharmaceutical industry, that will be beneficial. And most of these strains exhibit diversity in metabolic activity, and that is the part of Professor Mondal. We have isolated different microorganisms, and we have also tested for its probiotic activity, because there are certain characteristics of probiotic bacteria, whether those are present or exhibited by these bacteria or not. In that case, we have found that those are probiotic bacteria. But major drawback is there is high percentage of moisture as well as high percentage of salt. All of we know that if we prepare chana from milk, in that case, percentage of moisture is very high. Though percentage of moisture is very high, so its shelf life is very less. So it is conventional product. So people, they use huge amount of salt, because all of we know that salt, that is a preservative. So using natural preservative or class 1 preservative, they preserve this food item or cheese. So these are very much salty. So if we consume this cheese, before consumption, we should immerse in water for desalination. So there are two types of Bengal cheese. One is smoked and another is normal. And now cheese, already I have mentioned that cheese is nothing but chana. So processed chana. So you see, so first protein, that is coagulated from milk using different method. It may be by chemical method, that means using acid, organic acid, or by microbiological method. And this protein, this one is the common conventional method for cheese preparation. So coagulation, processing, molding, fermentation, curing, packaging, etc., etc. So there is a big list. And if we classify cheese, there are so many, more than 5 or 6 types of cheese are there. But if we commonly divide it into 3 categories: hard, soft, and very hard. So this Bengal cheese, this one is basically hard cheese. All of we know that churpi, that is available in hilly region, that is very hard in texture, and mozzarella cheese. So if we buy cheese from market, you see there are blisters type structure, that means hole. There are some holes. So these holes are generated by gas formation by different microbial activities. So due to fermentation and curing processes. And now you see those experts from DST who asked, your project proposal is of 18 lakh, but total market value is 20 lakhs per year. So how it is feasible? You see, potentiality, market potentiality of cheese, that is gradually increased year by year. So if we process this in scientific way, healthy way, in that case, it has also huge market potential. So before start our project, in that case, we should test different parameters. Those are basically physical parameters, sensory analysis, and proximate analysis. So physical parameter means that is pH, titrable acidity, syneresis. So in case of dohi or yogurt, there is one parameter, syneresis. That means that indicates water holding capacity. Water holding capacity basically. And sensory analysis, in first session, Dr. Rao, he has mentioned that sensory analysis is very much important. And from student point of view, all of we know that there is hedonic scale, 9-point hedonic scale. On the basis of hedonic scale, we can characterize sensory properties of any particular food product. And at the same time, suppose proximate composition. All of we know that proximate composition means basic composition, carbohydrate, protein, fat, moisture, and as indicates the mineral content. So these are the basic properties. And after characterization, these are the different parameters. They are weight, shape, size, density, pH, fat, etc., etc. There are so many parameters. And from these parameters, we can compare these Bengal cheese with other cheese. Those are available in local market. And sensory evaluation, you see, in case of sensory evaluation, they score on an average 7 to 7.5. Why not 9 or 9.5? Why not 9, 8, 7 or 7.5? Because this one is very salty. Due to this salt taste, that overall score that reduced to 7 to 7.5. And proximate analysis, you see, it contains huge number of minerals, vitamins, zinc, calcium, all of these essential minerals are present in this Bengal cheese. Now another parameter, that is the texture. Suppose one cheese or any food product that is very hard or very soft. So how can we predict or how can we evaluate that quality of the food product? Because we can say this product is very hard, but how much hard? Hardness is how much? That is determined by texture analyzer. So using a different probe, we can analyze texture, even chewing strength, gumminess, stickiness, all of those parameters we can determine from this texture analyzer. And we get different graphs. So from those graphs, we can predict what is the accurate quality of that food product on the view of textural property. And at the same time, using different scale, we can present in different modes. Now you see, it contains both saturated, unsaturated fatty acid, even trans fatty acid also, but little amount. Dr. Rao, he was worried that trans fatty acid that is detrimental to our health, but this one is natural product. It contains less than 0.001 gram per 100 gram, so very less. And monounsaturated fat, MUFA, PUFA, all fatty acids are there. And textural profile, these are the different textural profile: hardness, adhesiveness, springiness, cohesiveness, gumminess, and chewiness. And at the same time, if we take same picture, that means scanning microscopic picture, in that case, these are the these globules are present inside this cheese. Already I have mentioned that these type different natural product like this cheese also, it contain different number of various microorganisms present in the cheese, and these microorganisms are both of pathogenic category and non-pathogenic category or beneficial category. So our target was to isolate beneficial microorganism. And what is the microbial profile? Actually, that microbial profile that was characterized by lab of Professor Mondal at that time. And we have isolated these beneficial microorganisms by different tests. And these are the common microbial tests in microbiology lab. And our target was to isolate whether antimicrobial agent is present or not. You see, antibiotic can prevent the growth of different types of microorganisms, both gram-positive and gram-negative microorganisms. This cheese, it also contains antimicrobial compound. So these are antibiotic? No. These are the biopreservative. Basically, these are short-chain polypeptide. We have tested using E. coli and Bacillus subtilis. You see, these are the non-growth zone. So this indicates that this it may contain small chain fatty acid, and these are antimicrobial properties. And these are the microscopy of that microorganism. These are the microbial profile. You see, huge number of microorganisms are present in cheese. Yeast, aerobic, anaerobic, even you see E. coli, Clostridia, Listeria, Staphylococcus, all of these pathogenic microorganisms are present in this food product. Why? Because this is the conventional method. So, producer, they use empty hand. Their pots, those are also not sterile or properly sanitized. So, these pathogenic microorganisms are present. So, if we consume this product, in that case, we may affected by these microorganisms, pathogenic microorganisms. So our target should be remove these microorganisms during production, packaging, and preservation before consumption. And this is the diagram, basically how these are prepared from whole milk. So basic is coagulation, separation of chana from milk, then curing and selling that. So in this one is conventional natural fermentation process. So how it is possible? Because different microorganisms are present in milk, but during boiling process, those microorganisms, maximum microorganisms, removed from the boiled milk. But they use previous culture for curdling process, for chana making process. So previous culture that contains a huge number of required microorganisms. I may say required microorganism, not essential microorganism, required microorganism, because that culture may contain both harmful and beneficial microorganisms. So developed, these are the market available. This product is available in market, upper product, and this product, normal cheese and smoked cheese, these are prepared in our laboratory. That you can identify from shape. So these are the products developed by lab also from cow milk, and we have isolated microorganism from those Bandel cheese and used for the production of this product. And there are different possibilities of this work. So we have isolated that microorganism. So using this microorganism, we can develop different types of food product, and it may be used as antimicrobial agent as well as for yogurt production, for pharmaceutical purpose if it is feasible. Till now we don't know. And isolated microorganism that was used for yogurt preparation. Yogurt preparation and chana preparation. After coagulation, there is chana, and this chana is used for preparation of modified Bengal cheese in laboratory. You see, these are the some pictures, and this is the packaging system, right? Like Rasagolla, because Bengal cheese is preserved in 13% sodium chloride solution. So, if we use hot sterile 13% saline solution and packaged in this type of package, in that case, it will be preserved for more than 3 months. So our target was removal of these pathogenic microorganisms, isolation of different products which room temperature only, because it is preserved in 13% salt. So our target was preparation of cheese using these microorganisms under hygienic condition and preservation and removal of those pathogenic microorganisms. So after packaging, these products may be transported to different state in international market also. And there are different scopes, because you know churpi is the hard cheese that is available in hilly region. We have some scope with work with these churpi products with Professor Mondal in Leh Ladakh region also. So from that conception, we have dried this Bengal cheese using solar dryer. So drying cheese, that is also another possibility. And development of different probiotic food products and fermented production of bacteriocin. Because already I have mentioned that it contains antimicrobial agent, probably that is the short chain polypeptide. So if we produce and isolate those short chain polypeptide, after proper investigation, it may be used as bacteriocin. That means antibiotic-like product produced by bacteria. This one is the churpi, actually Leh Ladakh-based product. So our conception is production of churpi-like product from Bengal cheese using solar dryer. And we have designed one solar dryer, 3-layer solar dryer. And interesting fact is that we have designed this solar dryer for Leh Ladakh environment. So efficiency of any solar dryer, that depends on design criteria, length, width, etc., etc. At the same time, environmental condition. That means what is the relative humidity of air. Obviously, relative humidity of air of West Bengal, that is totally different from Leh and Ladakh. We have designed this solar dryer for Leh Ladakh region, and our target was maximum temperature should be 60 degree, above 60 degrees, 65 degrees centigrade. And at that environment, it was 62 degrees centigrade when their atmospheric temperature was 32 degrees centigrade. Their pollution load is very less, inclination angle that is very high, but in West Bengal, that is 33 degree, 23 degree. In that case, there is 33 degree. So intensity of light is very high in Leh Ladakh region. But last week we have tested that this solar dryer in this season also, when temperature is not proper, outside temperature is 27, 28 in Calcutta, but temperature of this inside of this chamber, that reaches now 60 degrees centigrade. So that is very good. Our target is drying of this product using this solar dryer. So I have mentioned just churpi that product, and you see what is the potential of dry cheese. In that case, we have dried that churpi of Leh Ladakh product, and after packaging, that local people, they sell in the Leh market. Value of that their local cheese value is 40 rupees per 100 gram, and after packaging, local people, they have sold at 50 rupees per 100 gram, and within half an hour, all packets that sold in Leh market. So there is huge potential, market demand, consumer demand. So our target is also dry product development from this Bengal cheese using this solar dryer. And these are some scientific communication. And thank you.
Thank you, Dr. Bera, for your brief, compact, but very appetizing lecture.
Thank you.
I apologize to everybody because we shall have one more lecture to make you more hungry and finally angry before food is served. Now the lecture is open for discussion. I request students to ask questions. But before that, I summarize what he discussed. At first, Dr. Bera highlighted how Bengal cheese is prepared. Then he talked about the composition of Bengal cheese, then instrumental analysis of the physical properties of Bengal cheese. Then he told about their investigations on the microfloral profile in Bengal cheese. And finally, he highlighted how to prepare new varieties of Bengal cheese using solar dryer. Now, any question from the students?
Sir,
Yes, please.
Good afternoon, sir. I am Kiya Das from Vidyasagar Institute of Health. Especially those who have lactose intolerance can consume cheese.
You see, cheese means chana that contains residual amount of residual part of lactose, because chana also contains your some portion of liquid moisture. Moisture comes from whey, so that whey contains also lactose. After preservation or curing, lactic acid bacteria, that lactose is converted to lactic acid by lactic acid bacteria. So if you consume just after curdling, in that case, it may contain some amount of lactose. But if you consume after curing, after fermentation process, auto fermentation process, natural fermentation process, in that case, it will be reduced or vanished.
Thank you, sir.
Any other question?
Hello, sir. I am Kanika Patro from Raja Narendra Khan Women's College Autonomous. Sir, my question is that we know pizza burger is junk food, but we also like to eat it. If we use Bengal cheese as a substitute of mozzarella cheese or Amul cheese, will it be somewhat beneficial for our health? What is your opinion?
Actually, your question is, if we consume Bengal cheese instead of
Mozzarella cheese or Amul cheese, any
etc.
other?
You see, there are more than 100 types of cheese available in world, and that variety depends on their manufacturing procedure, microorganisms used, whether it is cured or not, and different types are there. So variety means their taste, flavor, texture, those are different. So question of substitution is not there. So all cheese, different cheese, those have different advantages. Advantages in terms of your short chain polypeptide, microbial presence, etc., etc. So it is not substitution. We cannot compare with other cheese like that.
Okay, sir. Thank you, sir.
And one thing, cost of this cheese is very low. Per piece, per 50 gram, 6 to 7 rupees only. That is beneficial.
That you have told that the bacteria that is present, that is like bifidobacteria or lactobacillus, that is maybe carried out from the milk when preparing the cheese. But and that is you have analyzed by
No, sir, not only that. They are using old culture for curdling process.
Okay.
So old culture may also contain those type of microorganisms.
My specific question is that, other speaker also told, that our intestine contains, maybe those are the beneficial bacteria, but as intestine contains trillions of bacteria, so there may be some unculturable bacteria may be present, which may have also some beneficial effect. So during your analysis, that after preparation of the cheese, so have you analyzed by any like metagenomics or any kind of analysis?
No.
That what are the other, their percentage may be very small amount, but they may really also
they may
affect
contribute.
our system. instead of just a few microbes that which we known microbes like bifidobacteria or lactobacillus, that which all we consider as a probiotics. But as Professor Mondal also told, that it's a personalized medicine or personalized probiotics to take it. But we are not really analyzing in those cases, like Professor Mondal told that.
Yeah, that is true. That is true.
Started analysis, so that's why I just want to.
There is huge scope.
Yeah. Okay, thank you.
Yeah, thank you. হ্যাঁ কি একটা করছি হ্যাঁ বলো
Sir, Kothakoliide, Midnapore College. I have a question. Sir, we generally tells people to take less fat. On that context, can you tell us about nutritional importance of Bengal cheese?
less fat content হ্যাঁ fat content is very less major portion is cheese means ছানা protein content
The nutritional importance.
Nutritional importance you see, so obviously you know the importance of protein in our diet. At the same time, protein is disintegrated in our gut by enzyme or different means, so it converted to short peptides, small chain peptides, then amino acids, short chain peptides, different products and derivatives are there. So in cheese already decomposed some fraction of protein that is not decomposed, that is splitted into short chain polypeptide, amino acids like that. So that is easily digestible than normal cheese or chana.
Thank you, sir.
Okay, sir. We extend our sincere thanks, though you have forgotten to bring some sample of Bengal cheese for us. Now, I would like to invite the final speaker before lunch, and he is a renowned professor, Professor Ananta Kumar Ghosh, from the Department of Biotechnology of Indian Institute of Technology, Kharagpur. The research output of Professor Ghosh is prolific, and before I summarize that, our students would like to felicitate Professor Ghosh. Professor Ghosh is a versatile genius. His research interests circle around molecular virology, seri biotechnology, hybridoma technology, cell and molecular biology, recombinant DNA technology, gene expression, antimicrobial proteins and peptides. Currently, he is interested to explore the molecular organization of virus infecting the Tosher silk moth, Antheria mylitta. Professor Ghosh was a postdoctoral fellow in the Department of Pathology, University of Southern California School of Medicine, Los Angeles, California. He was also a postdoctoral fellow in the Department of Cancer Biology, Harvard University School of Public Health, Boston. He was a research associate in the Department of Microbiology and Immunology, Stanford University School of Medicine, Stanford, California. He was also an assistant professor of research pathology in the Department of Pathology, University of Southern California School of Medicine, Los Angeles, California. He also acts as a visiting scientist in the Department of Microbiology, University of Washington at Seattle. He has more than 70 publications to his credit in different international peer-reviewed journals. He has already utilized research grants from different agencies of Government of India. As many as 16 such projects have already been completed by him, and 4 are in progress. He has guided 2 postdoctoral fellows and guided 12 PhD theses, 28 MTech theses, and 24 BTech dissertations. I welcome Professor Ghosh with his deliberations.
Thank you, sir. And also respected delegates and student friends. I want to thank the organizer for inviting me to deliver the lecture on this topic. Actually, let me apologize myself that I do not directly work on the topics of the seminar that is going on today here, that food informatics. Although it's not true that I did not work, last few years I did work on this food microbiology a little bit, but not that extent. That's why I am not delivering that lecture on the topics. Actually, my topics was, what I work is actually that stored food grains that was present in different warehouses, mostly the fungus that affects the stored wheat, and then how to easily detect them. So developing some detection system. But anyway, I am not talking on that, but my other area of research, as Professor has mentioned, that lately I worked a few years on biomaterials. And that's actually, I'll be talking in last maybe 15 minutes or so quickly. I know that this is, the lunch is getting ready and everybody is waiting to have the lunch. So just take a little bit patience to listen it, although it is a little bit not related to the food. Okay, anyway. So my topic is today is in vitro and the in vivo evaluation of non-malveric 6-session-based hydrogels in skin regeneration. So before I start it, let me know what is the skin. Skin, you know, it's the outermost organ of our body. And since skin is the outermost organ of our body, it is always prone to damage. And that's why it always needs the repair. And damage may occur by infection, burns, accident. Okay, and that's why you always need the repair of the skin. And to repair the skin, we always need a scaffold that if skin is lost, so we need a scaffold which will help to regenerate the skin. So actually our research work is to develop some scaffold which will help to repair the skin if the damage is occurred. But in the market there are several scaffold is available, but that is sometimes costly or made up of some synthetic materials which is not easily biodegradable. So we are using some natural resources which will help to prepare this scaffold. And we are using this natural source at various natural sources, and one of the natural sources that are using it is the silk. Silk, you know, is mostly used for making the fabrics, and in India we prepare the silk mainly 2 kinds of silkworm. One is Bombyx mori, that is mostly majority of the silk is prepared, but another variety is the non-malveric silkworm which produce the Tosor silk or the Muga silk or other kind of silk. So our work, mainly this eastern region, actually we grow a lot of amount this Tosor silk. So we are using this Tosor silk protein, it's a natural source of protein, to prepare the scaffold which will support the regeneration of the skin. Okay, so this is, I mentioned it, that mostly I am using this Antheria mylitta, which is this non-malveric silk, and this is its cocoon. From here actually we obtain the silk fiber. So if we look at the silk fiber is made up of mostly 2 kinds of proteins. One is these 2 major proteins, it is the fibrin protein, and then it is wrapped by a glue protein, that's called the sericin protein. Okay. So when people extract this fiber, they mostly in the industry, they remove this outer coat protein, that's called the degumming. So we are using actually these waste materials from the silk industry that whether we can use that as a biomaterials, natural biomaterials to prepare the scaffold. Because this sericin pupillage work and the size is a protein, the size is about 30 to 200 kilodalton, and then it has various antioxidant, antibacterial, UV resistance, and the anti-apoptotic properties. So therefore we select this protein and, to be used for the skin tissue regeneration with this scaffold. Okay. So our objectives is the construction, characterization, in vitro and the in vivo evaluation of this non-malbary sericin protein, along with the chitosan, which is also a natural compound available from various crustaceans, and then use as a hydrogel as a dermal substitute. So this is some commercially available hydrogels that is available in the market. So hydrogels is basically a network of hydrophilic polymer. It produced through chemical or the physical cross-linking. It is highly absorbent and is flexible in nature. So we started our work with this sericin. We took this silk cocoon, cut into pieces, and then we have extracted it because it's a water-soluble compound. If we autoclave it in the water, we get the sericin solution. Then we dry it to make the sericin powder, and then this sericin, then again rehydrated it with the water, so make the sericin solution. Then we have equal amount of sericin and the chitosan, another naturally occurring biopolymer, mixed it and then cross-linked with the genepin to prepare the gel or the hydrogel. So then we check this property of the hydrogel, that hydrogel should be porous because you want it to grow the cells there, and we show that the porosity about 75 to 90%. And then after preparation of the hydrogel, we check that what's the pore size. So we did the scanning electron microscopy, and when we prepare the hydrogel, we take different ratio of the sericin and the chitosan, and this is the different ratio of the chitosan and the sericin, and then we find out that this is the ratio, means twice chitosan and one ratio is the sericin, and is the best one, because it has the pore size also 57 nanometer, micrometer, because the pore size should be enough that the cell should go through it for its growth. And that is we did by the scanning electron microscopy studies. The interconnection between the pore is there, and the fabricated hydrogel after preparation, this is stable up to 6 weeks. So after preparation, we can use up to 6 weeks for that, allowing the growth of the cells. Then after preparation of the hydrogel, since the biomaterials, we have to study what is supports the growth of the cells. So we took some human dermal fibroblasts because we wanted to use the skin regeneration. So we took the human dermal fibroblasts, then we started the growth curve using that hydrogel to grow the cells. And as you can see is that up to 30 days, the cells is growing very happily, and this is one kind of assay by which we study the growth of the live cells. And not only it supports the growth of the cells, we have to see that whether that matrix that are using it in the body, that's also biocompatible, that it should not lyse our blood cells. So we have started taking that human RBC, that is a control, just positive control, just the water which will lyse the RBC, but none of this hydrogels is lysing the RBC. Then also anything we use in the body, that should not be any immunogenic, that immunogenicity should be very less. So after culturing this human derived macrophages in that hydrogel, we have studied the inflammatory cytokines like IL-1 and this TNF alpha, and see this the positive control where both these things are increasing the level, but then in the hydrogels that are minimally this inflammatory cytokines is produced, and that indicates that this is non-inflammatory in nature, this is non-toxic, as it is the biocompatible. And then when we are studying the growth of the cells, this is the electron microscope picture to see that inside the hydrogel the cells are growing, and then how deep the cells are growing. And then we did some confocal microscopy, staining the cells with rhodamine and the phalloidin, and this is the scale. Then if the cell is going deeper, then it is taking yellow color. If the cell is in the surface, that is taking this blue color. So in this ratio, the 2 chitosan and the 1 sericin, this is the most appropriate mixture of the chitosan and the sericin, which is supporting the growth of the cell not only in the surface of the hydrogel or the scaffold, but it's going inside in the matrix.
and that is the cell individually, if I see it by staining it, also with phalloidin, and which is staining the nucleus as with the cytoskeleton. And the blue color is the nucleus and the yellow color is the cytoskeleton. Then we can see the cell is growing very happily. So this hydrogel is supporting the uniform on the deeper migration of the cells, and the microscopic image is showing that the cells are proliferating with the podia and interconnecting with each other. Okay. Then we are using that, since the skin is made up of not only one kind of cells, various kinds of cells are there. So we are trying that whether it can support the growth of at least two kinds of cells. So we are taking this human dermatofibroblasts as well as the keratinocytes, and the color did not come very well, but actually there are two kinds of cells are there. And then when I see that after seeding it, that both the cells are seeding very nicely, and this is also the electron microscopic picture to see that the cells after seeding both the cells, the cells are the color, red color is not coming anyway. So the red color which is present in the lower side and in the upper side is the keratinocyte. Both cells are attaching into this scaffold. And then when we study that co-culture, I mean both kinds of cells over the period up to 21 days, as you can see that day 7, and as the cells time is going, the cells are growing very densely inside the hydrogel. Okay, supporting that our hydrogel is supporting the growth of the cells in vitro. Then we have after supporting the growth of the cells, we are studying that when the cells is growing, and then to growth of the cells very nicely, it should take the support of the other cells also. So to check the other cells, so cell always produce some extracellular matrix protein. So the cell and cell will adhere very nicely. So we are studied by doing this quantitative RT-PCR, semi-quantitative RT-PCR, the collagen, then metallomatrix protein 9, metallomatrix protease 2. And we have seen it that this hydrogel is supporting this production of the extracellular matrix protein, and so that other cells will grow very happily. And then in addition to these hydrogels, in addition to the ketosans and the sericin, we also incorporate another compound, this glycosinoaminoglycans. And addition of these glycosinoaminocontents is also support the growth of the cells very nicely. In addition, we have also had some growth factors. So in addition of the sericin, ketosan, glycosinoaminoglycans, and the growth factor, they support much more the growth of the cells as well as the production of the extracellular matrix protein. And then after studying the effect in vitro, and then really to see that whether it works in vivo, then we implanted the hydrogels in the rat skin, making little bit incision in the rat skin, then we put the hydrogel. So this is the picture after making the incision, then ultimately after putting the hydrogel inside, and then we are stitching it. Okay. And after doing that, so we put that hydrogel in the rat skin for about 28 days, and after 28 days, then we are seeing that whether the hydrogel is supporting the growth of the cells there. Then after making a slicing over there and doing this histologic analysis, we are seeing that it is supporting the growth of the epidermal tissue, growth of the hair follicles, as well as the lot of collagen bundle. In addition, only the ketosan, which is supporting in respect to that control ketosan, in respect to that when you are using the sericin as well as this ketosan, then it is the collagen layer is getting much more thicker, or it is supporting the growth of the hair follicles. Then we have studied this, whenever there is a new growth of the cells in anywhere where implanted tissue, to support the growth of the cells, usually there is also growth of the new blood vessels takes place, so that when the cells are growing up over there, the nutrition will go through it. So after implanting these hydrogels into the skin, and then we have also studying the whether it is supporting the growth of the new blood vessels. So now one of the markers to analyze the growth of the new blood vessels is the CD31. So as you can see, compared to the control and only ketosane, then we are seeing this ketosane and the sericin containing hydrogel, it is supporting growth of the more blood vessels. Okay. So that's also indicate that our hydrogels that we are preparing it is supporting the angiogenesis. Angiogenesis is the development of the new blood vessel into the skin tissue. Then after putting the hydrogel, also we have to see the inflammation, means whether it's the immunologically how much it is supporting, means whether it is immunogenic or the non-immunogenic, which is the foreign substance. So we have after putting the hydrogel, we are starting these products on the interleukin, TNF alpha, as well as they are the pro-inflammatory cytokines, and the anti-inflammatory cytokines IL-10, the interferon gamma. So as you can see, the anti-inflammatory cytokines is producing much more, but the pro-inflammatory cytokines is producing very less. And this indicates that our hydrogel is minimally immunogenic, because due to surgical procedure and some immune response might have produced the body. Otherwise, the hydrogel is non-immunogenic in nature. So in summary, so we have prepared a strong porous hydrogel by mixing the sericin and the ketosanes. The fabricated hydrogel is cytocompatible because it supports the growth of our cells, viability, distribution inside the hydrogels, and when the cells is growing, it is interconnecting each other and supporting the growth of the human dermal fibroblasts. Then also it supports growth of the two layer of the cells, that is the fibroblasts as well as the keratinocytes, and also it enhances the production of the extracellular matrix component, so that it will support the growth of the other kind of cells. When we studied in vivo, we have seen that the hydrogel is supporting the reconstruction of the skin tissue. It is helping to develop the hair follicle, dense collagen, and also the natural mature blood vessels. So in conclusion, that our sericin ketosane containing hydrogens are cytocompatible, means supports the growth of the cells. It is non-hemolytic, it's a biocompatible, non-immunogenic, and exhibits a good prospects for a natural dermal substitute in vivo for skin regeneration. So these are a couple of publications which came out from this work. And then two PhD students who actually did the work in collaboration with my two other professors, and DST and DBT for funding support. And thank you very much.
Thank you very much, sir, for your very important and fascinating lecture. You explained how the way you fabricated hydrogel using sericin protein of silk moth, Tosher silk moth, and ketosane. And then how you analyzed biocompatibility of the fabricated hydrogel in living being. It was found to be safe, both immunogenically and from other standpoints. And finally, you analyzed the prospect of the fabricated hydrogel in remodeling of damaged tissue. You made both in vitro studies with human fibroblasts and also in vivo studies in rabbit model. I have got a small question.
Yes.
You mentioned that this fabricated hydrogel lasts for only 6 weeks. Then is it very much commercially viable, or what do you do to strengthen its longevity? Or
since this is that initial work, we may have to use some other preservatives or so, because the natural substance, protein mostly based.
now, are you trying to increase
Yes,
its
yes, to increase.
longevity?
Like initially we used only ketosanes and then the sericin. Then we have used glycosaminoglycan. Then we use the growth factors. Later on we have used also the antibiotics, supplementing. So
You're
that
getting some good results?
good results, yes, yes. So further work is going on in this.
Any other question from anybody else? Yes, please.
Yes.
Hello, sir. My name is Kanika Patra. I am from
Louder.
I am from Raja Narendra Lal Khan Women's College, Autonomous. Sir, my first question is that The hydrogel prepared by you, is it available in the market?
No, it is not yet available in the market because we have just prepared it. Coming anything in the market, it takes a long process.
Or my second question is that in case of xeroderma pigmentosa, is it beneficial?
Xeroderma pigmentation is a genetical disease. Actually, it's actually a defective in that basically the repair pathway, the DNA repair pathway. So one of the genes is defective there. That's why xeroderma pigment. There are 7 genes are there for the xeroderma pigmentation. And if any genes are defective, then if our DNA get damaged, then it cannot repair. That's why the disease here. Here it is not you can apply there. If you're here is that's the genetic defect. So this is not a genetically.
So in case of skin redness, we can apply this.
Yes, if there is skin damage, anyway, if the skin is get damaged, so to quickly repairing the skin, then we can use that as a scaffold so that it will support the growth of the cells. That's we are studying it. That we have studied in vivo also. They're making the lesion in the skin, then we are putting the hydrogel, then it is supporting the growth of the cells.
Okay, sir, my third question is that it is totally organic, mineral oil free, paraben free, silicone free, or animal cruelty free.
Yeah, it doesn't have any other except natural substances. Even the genipin that we are using it for cross-linking, that is also from the plant source. So there is no other artificial or that synthetic chemicals is not there.
Sir, we will hope that you bring the product as soon as possible.
Thank you.
Any other question?
Other no, because this forum is actually for food. I should have maybe some other occasion since this department is here. Food informatics is there.
Thank you, sir.
Okay.
And we are now closing the pre-lunch technical session. And I request everybody to move to the canteen for having their lunch. And try to return as quickly as you finish your grab, because we shall have another 2 rounds of scientific discussions followed by cultural program.